Okay, so here we are going to go over the mechanism for how RuBisCO is responsible for carbon fixation. Now, before we jump into the mechanism, we need to understand what is RuBisCO, where is this mechanism occurring, and what is the purpose. So, RuBisCO is an enzyme. It stands for ribulose 5 bisphosphate carboxylase oxygenase. Carboxylase and oxygenase means that this enzyme can both carboxylate and it can oxygenate. Now, the ability of RuBisCO to oxygenate is actually one of its limitations because the whole purpose of RuBisCO is a key step in the Calvin cycle, which is carbon fixation. You want to add carbon dioxide to ribulose 1,5-bisphosphate to generate 3-phosphoglycerate. But if we are oxygenating, we cannot carbon fixate. Essentially, when RuBisCO is surrounded by a lot of oxygen, instead of carboxylating, it's going to oxygenate. So this is a limitation of RuBisCO because in effective Calvin cycle, in the Calvin cycle effectively occurring, we want carboxylation, not oxygenation. This brings me to another point, which is that RuBisCO is actually the most abundant enzyme in the entire world. Number one being that it is inefficient. It has the ability to oxygenate, and we don't want that in the Calvin cycle. So that's why we have more of it. Second reason why we have lots of it is because it has a very slow turnover rate. And then the third reason, and the most obvious reason why it's the most abundant enzyme in the world, is that it is because it is found in plants. And plants are everywhere in the world, and they're all undergoing photosynthesis and the Calvin cycle. So we have to remember this is the most abundant enzyme in the world. Now, once again, where is RuBisCO being used? Well, we have the Calvin cycle, and the Calvin cycle is also referred to as the light-independent reaction. Remember that when we talk about plants, we have photosynthesis, which generates ATP and NADPH, and those ATP and NADPH are going to be used in the light-independent reactions, which is the Calvin cycle. Now in the Calvin cycle, we start off with ribulose 1,5-bisphosphate, depicted right over here. And what we're going to do is we're going to introduce this carbon dioxide group, and we call this carbon fixation, and we're going to generate 3-phosphoglycerate, which is going to be reduced into glyceraldehyde 3-phosphate, G3P. And then this G3P, it'll have various fates, but we're not going to get into its varying fates. That's for a separate video. Now that we've oriented ourselves on what RuBisCO is and where this reaction is occurring, let's look at the mechanism. Once again, the purpose of RuBisCO is going to be attaching the carbon dioxide to ribulose 1,5-bisphosphate, and this is called carbon fixation. Okay, so here we are looking at a zoomed in section of our enzyme, and our enzyme is RuBisCO. Now, enzymes are made up of amino acids, or they're made up of proteins. So specifically in the active site of RuBisCO, we have these amino acids of interest. We have histidine, we have glutamic acid, we have aspartic acid, and then we have lysine. And at the center of all this, we have a magnesium ion. Now, the function of magnesium is really important because magnesium is responsible for having these interactions with the substrates to orient the substrates in a proper manner. And magnesium also mitigates that negative charge. We can see that aspartate and glutamic acid actually exist in their deprotonated anionic state. So magnesium 2+, since it has that positive charge, it's mitigating negative charge, and it's just orienting all of our substrates. Now, what are our substrates? Well, we have ribulose 1,5-bisphosphate because remember, that's what we are trying to add that carbon dioxide to. And then we also have lysine, but this is a very interesting lysine because it has been carbomoylated. Essentially, we took carbon dioxide and we added it to the end of lysine. Now, when the lysine is not carbomoylated, the enzyme is inactive and this process will not be able to occur. Now, that is a regulatory mechanism, and that's for a separate video as well. Once again, here we're only going over how carbon dioxide is attached to ribulose 1,5-bisphosphate. But it's important to remember that the carbamylation of lysine is very necessary for this reaction to occur. Now, once again, these are our amino acids of interest. Magnesium is orienting us, it's acting as a cofactor, and then we have our ribulose 1,5-bisphosphate. Now, in the first step of this reaction, we see the importance of the carbamylated lysine. Essentially, it acts as a nucleophile, and it's going to abstract the hydrogen that is found on the third carbon over here. When it abstracts that hydrogen, these electrons will go over here, and essentially these two carbons will form a double bond with each other. And that's really important to create that double bond because we're going to create an ene, and it's going to become more reactive and it's going to act as a nucleophile. So each step that we do in this reaction is preparing it to become more reactive. So once again, we see the importance of the carbamylated lysine. It abstracts that hydrogen. As a result, these two carbons will now share a double bond. But now remember, carbon can only have 4 bonds. If we form a double bond over here, this carbon is going to have 1, 2, 3, 4 bonds. But this carbon has 1, 2, 3, 4, and then 5. Carbon can't have 5 bonds. It's not stable that way. So it needs to lose a bond, and it's going to lose a bond right over here, in which these electrons are actually going to be passed over to this oxygen. And we see that in this step over here because we've created an enediolate intermediate, which is much more reactive than our ribulose 1,5-bisphosphate. So this lysine, the carbamylated lysine, has abstracted that hydrogen. It's depicted in yellow. And then we can see that double bond now formed between the two carbons. That is our ene. Now, this was the oxygen that gained those electrons that were once shared between the carbon and oxygen. And as a result, this oxygen has picked up a hydrogen. Because once again, this reaction, it's occurring in a fluid-like space where we do have hydrogen present for protonation and deprotonation. So this oxygen has picked up a hydrogen. Now, the next step is that we actually introduce the carbon dioxide. Because remember, the whole purpose of this mechanism is to attach this carbon dioxide to ribulose 1,5-bisphosphate to generate 3-phosphoglycerate. So let's take a look at how this carbon dioxide is incorporated. So now we see the importance of histidine. Now, histidine, nitrogen has these 2 lone pairs. Now, this nitrogen is going to act as a nucleophile, and it's going to abstract the hydrogen over here. Now, when it abstracts that hydrogen, these electrons will go over and be shared between this carbon and oxygen. Now, once again, we have to remember the key rule where carbon has 4 bonds. Now, if it forms a bond over here, it has to lose a bond somewhere else, or else it's going to have 5 bonds. And that's essentially where this ene intermediate comes into play. Now, this enediolate intermediate, it's much more reactive than our ribulose 1,5-bisphosphate, and it's going to act as a nucleophile and go over and attack this carbon. Now, why is it able to attack this carbon over here? That is because this magnesium over here is interacting with the oxygen. And essentially, it's polarizing this bond, making carbon more susceptible to being attacked by that enediolate intermediate. So now the enediolate intermediate attacks this carbon dioxide because this bond has been polarized. And remember, once again, carbon dioxide can only have 4 bonds. So as a result, these electrons over here are going to be passed down to this oxygen. Now, if we take a look over here, we can see the carbon dioxide has been incorporated to our molecule, which was once ribulose 1,5-bisphosphate, But now, once the carbon dioxide has been added, we refer to this as beta keto acid intermediate. Now, let me just get rid of this because this is from the previous one. The Enediolate was the more reactive version. Now, taking a look, once again, we have our carbon dioxide incorporated. This oxygen bears a negative charge because it used to share electrons with the carbon, but now those electrons have been passed over. We can see that histidine, remember, it abstracted that hydrogen, so it has a plus charge now. And we have our beta keto acid intermediate. Previously, we had that enediolate intermediate, which was reactive and acted as a nucleophile. Now, the next step is a hydration step, and this hydration step is really key to creating a very unstable intermediate, which is going to break apart into two 3-phosphoglycerate molecules. Now, how does this hydration step occur? Well, we have water. Now, once again, oxygen, it has these lone pairs. It's going to act as a nucleophile once again and attack this third carbon. When it attacks that third carbon, remember, carbon can only have 4 bonds. It must lose a bond, and this time it's going to lose it to this oxygen over here. Now, this oxygen will gain a negative charge. Once again, we can understand why this carbon is being attacked because this magnesium is interacting with the oxygen, once again polarizing the bond, and then oxygen is able to act as a nucleophile and attack the carbon. When it attacks the carbon, we can make note over here that this oxygen gained a negative charge because those electrons were passed over. And now water, this oxygen formed a bond with the carbon. We lose a hydrogen in the process because remember, originally we had H2O. One of the hydrogens is lost. We have this OH. This is our hydrated intermediate. This is a very unstable intermediate, and it's just going to fall back on itself to split into 2 different molecules. So when it falls back on itself, essentially these electrons over here are going to be passed over and be shared between the carbon and the oxygen. And as a result, remember, carbon can only have 4 bonds. It must lose a bond. So it's going to lose its bond right over here to this carbon. And these electrons are going to be passed over to that carbon. Now this over here is our first 3-phosphoglycerate molecule. We can make note of that right over here. This is our first 3-phosphoglycerate molecule. Where did this oxygen come from? It came from that hydration step. It's really important to keep track of where things are coming from. Now, once again, we're left over with this. This is our carboanion intermediate because this carbon, remember, it gained those electrons from the carbon, and that carbon was free to go in the 3-phosphoglycerate. Now, how do we mitigate this negative charge? Well, we can add a hydrogen because a hydrogen has a positive charge. Where is that hydrogen going to come from? It's going to come from that carboxylated lysine, which had abstracted the hydrogen in the beginning. It's going to come back and it's going to be given to that carbon. Now carbon, it's going to act as a nucleophile. It's going to abstract that hydrogen from that carboxylated lysine. And as a result, hydrogen has been added. We got our 3-phosphoglycerate molecule. So we got 2 3-phosphoglycerate molecules from one ribulose 1,5-bisphosphate. Now the 3-phosphoglycerates that we produced, they have varying fates. They can undergo glycolysis. They can go on to produce starch or sucrose. But first, of course, these 3-phosphoglycerate molecules must be reduced to G3P in the Calvin cycle. Now, once again, we're going to quickly go over this one more time to refresh our memory of what's happening. We have our enzyme. What is our enzyme? It's RuBisCO. Enzymes are made up of proteins. Proteins are made up of amino acids. And we have these amino acids of interest that are found in RuBisCO: histidine, glutamic acid, aspartic acid, and carboxylated lysine. And they're all interacting with our magnesium ion. And then we also have ribulose 1,5-bisphosphate, which is also interacting with our magnesium. And the whole purpose of magnesium is to balance the negative charge and orient the substrates. In our first step, The lysine, the carbomoylated lysine, acts as a nucleophile, abstracts the hydrogen, which results in our enediolate intermediate. Our enediolate intermediate is much more reactive and can act as a nucleophile, and we can note over here that the hydrogen has been abstracted by the carbomoylated lysine. Now carbon dioxide enters the mechanism, and the enediolate will act as a nucleophile and attack that carbon because this bond has been polarized by the magnesium, making the carbon more susceptible to nucleophilic attack. And the histidine will abstract the hydrogen. Now, after that, we've created our beta keto acid intermediate, and it's followed by a hydration. A hydration will introduce the OH group, and that OH group, once it's incorporated, we have our hydrated intermediate, which is highly unstable, and it's just going to fall back on itself to create our first 3-phosphoglycerate and our carboanion intermediate, which simply just has to pick up that hydrogen from the carbomoylated lysine, and as a result, we get 3-phosphoglycerate. Now, that is the mechanism for how RuBisCO carboxylates. Going back up here. So we have just completed this first step, which is carbon fixation. To complete the Calvin cycle, we have to undergo reduction and regeneration. And that is essentially the RuBisCO mechanism.